Bharat, can you hear me? Bharat,
Hello.
Bharat, can you hear me?
Yes, sir, I can hear you clearly.
Can you just WhatsApp the CV? Somehow I've lost it. Send it to
Okay, sir.
drdamle@gmail.com, so I'll read it.
Sure, sir.
Is Manigaru there? Yeah. Subramania ji. Bharat, received. Thanks.
Hello.
Yeah.
Received.
Sir, I had sent you through Dr. Damle@gmail.com.
Received.
Thank you, sir. Hello, Dr. Wangala.
Yes.
Am I audible to you, sir, clearly?
Yeah.
Okay, sir. Thank you.
Mani ji, namaste.
Namaste.
All guns blazing.
Huh?
All guns blazing.
Yeah.
You're always.
Yeah.
I think you have met or spoken to Dr. Shiban. Shiban is here. Dr. Shiban?
Sir, Shivan sir has not yet joined.
Oh, he has not yet joined. Dr. Manoj?
Hello.
Yeah, Dr. Manoj, it's a pleasure introducing you to Subramania Vangala.
Hi, sir.
Yeah, he's
Hello.
popularly known as Manigaru.
Hello.
He is popularly known
Hello.
as Manigaru. Mani Garu. Garu is, the respectable word in Telugu.
Right.
Money only.
Like Chetan.
Okay.
Mani Chetan is Manigaru in Telugu.
Okay.
Subramani is Mani.
Okay, sir. Nice talking to you, sir.
Yeah, nice talking to you, Dr. Manoj.
And who else is there?
Dr. Shaiju is there. Dr. Shaiju.
Yeah, introduce
Hello,
yourself.
sir. Hello, sir. Hello, sir.
Hello,
Dr.
Shaiju. Get introduced to Dr. Subramania Vangala. I think you should take his number because he will be of use for your PhD and other thesis. He is a very resourceful person.
Thank you. Hello, good evening, sir.
Good evening.
Huh.
How are you?
Even though I am in Ayurvedic fraternity, I am doing my research in molecular genetics in veterinary university.
Uh-huh.
My work is on hepatocellular carcinoma. I am doing work.
Okay. Trying to discover
So,
some herbal Ayurvedic drugs?
yes, sir. Actually, the drugs, traditional medicines, I am trying in hepatocellular carcinoma, where including the transcriptome analysis. It includes my transcriptome analysis also. So, So anyway, I think later I can, in detail, I can brief my work so that maybe it will be certainly, as Dr. Damle told, it will be helpful for my work also.
Yeah.
And so I would like to introduce Dr. Sadat Nagar. He is a secretary of our association, Ayurveda Medical Association of India, Kerala.
Hi.
Hi. Hi, sir. Good morning. Hello. Good evening. Good evening, sir. How do you feel? Hello. Can you hear
Yeah.
me?
Good. Speak a little closer to the mic because I can't hear you much.
Sir,
Good evening. Good evening, sir.
yeah, it's not so audible, it seems.
Okay, maybe they hear the Manigaru. Sadat is one person who can give you competition in your modeling as a hair model.
And Dr. Vishnu, sir, he is an associate professor from Department of Ayurveda, Shantigiri Ayurveda College.
Okay.
Dr. Vishnu?
Hello, sir.
Hi, Dr. Vishnu.
Yeah, myself. Am I audible?
Yeah, you're audible.
Okay, sir. Good evening, sir.
Good evening. Yeah.
Yeah, Manoj, who is going to give this one?
Yeah, welcome speech by Dr. Ajit Anand. He is our former General Secretary, and presently he is the Vice President of Travancore Kochi Medical Council.
I will just introduce you and then you take over.
Okay, sir. Okay.
Yeah. Dr. Shiban, are you there?
Hello. Hello, sir. Dr. Shiban will join in the meantime. Okay, so he has asked me to start the meeting and he will join, sir.
Okay.
So good evening, everyone, and we are going live on YouTube, and right now we are live on YouTube, and over to Dr. Damle, sir.
Yeah, this is one of the successful series we have been having, and we have been having audience of up to 1,000 people in various platforms, 2 YouTube channels, 2 Facebook channels. Then WebEx is only a closed group. So I request Dr. Manoj to start the proceedings today. Thank you very much.
Good evening, one and all. We'll directly go to the program. So I would like to invite Dr. Ajit Anand, who was a former General Secretary of Ayurveda Medical Association of India and former ex-CCI member and presently Vice President of Travancore Kochi Medical Council, Kerala. I invite Dr. Ajit Anand for welcome speech.
Am I audible?
Yes.
Yes. Good evening, friends. Good evening, doctors. And this is a very auspicious occasion. I have some network problem is here, so sometimes this voice may be interrupted. I think I am audible now.
Yes. Yes.
Yes.
Okay.
Please
Okay.
continue.
Thank you, Dr. Damle. Yes, AMA is conducting so many programs to enhance the clinical skill, research skill of Ayurveda graduates and postgraduates throughout the globe. As a part of this program, as a part of this continuing program, this time we have association with Hetrimed Pharmaceuticals and Ayurveda Vilasini Vaidyashala to conduct such a program and an hour with expert. And this time luckily we got a great personality from the field of bioscience, Dr. Subramanya Vangala, to present this program today. He's a renowned pharma scientist and expert in the field of drug development, regulatory matters of drug, as well as toxicology and clinical pharmacology. With great happiness, I welcome on behalf of Ayurveda Medical Association of India, Dr. Subramanya Vangala to this program.
Thank you.
And next, I welcome Dr. Rishikesh Damle, who is MD of Hetrimed. He is not only an entrepreneur, he is a real researcher from this field of Ayurveda fraternity. Personally, he is working on drug development for COVID-19. Welcome, Dr. Damle.
Thank you very much.
Then Dr. Manoj Kalur, he is our family member. He is a leader of Ayurveda Medical Association, former vice president, as well as former CCM member. He is leading Ayurveda Vilasini Vaidyashala. And welcome, Dr. Manoj Kalur, for this program. And Dr. Madhu is here. He's a role model of teachers. He's working at Kottakkal Ayurveda College, and he's a HOD of department. He's working as in department of Driveguna in Kottakkal Ayurveda College, and I think his presence will be a great help to enrich this program. Welcome, Dr. Madhu. And Dr. Vishnu, who is working in department of Agadandra Shantigiri Ayurveda College, Palakkad. He is also here as an expert of this subject. I welcome Dr. Vishnu for this program. Then I welcome Dr. Sadhu Dinagar, our beloved General Secretary, who is leading Ayurveda Medical Association of India now. He is the main organizer of so many continuing programs which are happening in this COVID period. Welcome, Dr. Sadhu Dinagar, for this program. And I welcome CEO of Atimet, Dr. Lata, is the wife of Dr. Rishikesh Damle. She is also an Ayurvedic doctor. Welcome, Dr. Lata, for this program. And I also welcome Dr. Shibin Kanchu. He's basically in USA. He's a gastroenterologist and chairman of Atimet. On behalf of Ayurveda Medical Association, I cordially welcome Dr. Shibin Ganju for this program. Then I welcome Dr. Shaiju. He is an expert from our field, from Ayurveda fraternity. He is conducting so many research on immunology as well. Dr. Shaiju, I welcome you for this program. And I welcome all the delegates and the participants attending this evening for this program. And I think we will have a wonderful session today. Thank you.
Thank you, Dr. Rajit. So let me invite Dr. Rishikesh Damle to introduce Dr. Subramaniam.
Thank you very much. Dr. Subramania Vangala, as I told earlier, is popularly known as Manigaru, is actually the real guy who has been deep into research as far as the drug development is concerned. He, in a nutshell, that he's a guy who has been there before a drug became a drug. That means during drug development and FDA approvals, including investigational new drug applications, NDA, he has been there everywhere. He has experience in both toxicology as well as efficacy studies. So briefly, his CV is pretty big, and his brief CV itself is pretty big. To introduce right now, he's working as a co-founder and CEO at Reagin Biosciences, which is based in Bangalore and Hyderabad. He has got 25 years of leadership experience with increasing responsibilities. He has a PhD in the biochemical mechanics of chemical carcinogenesis from the Department of Biochemistry. Now you know where he's coming from. That is in Canada, Memorial University, Canada. He conducted his postdoctoral research in molecular toxicology at School of Pharmacy, University of Colorado, USA, and continued as a specialist scientist at School of Public Health, University of California, Berkeley. If you look at his industrial experience, he has worked in toxicology, PKPD modeling, more than 30 IND applications, 7 NDA, ANDA applications. He was part of development of Zalepon, it's a short-acting sedative hypnotic, Tigacel, then Canagliflozin, then Tipso, etc. So basically, he has insights and real insiders experience apart from knowledge of drug development, and his focus is on toxicology. Now he has around 58 publications in very renowned international journals, and large work experience in private as well as academic institutions. With this brief introduction, I request Dr. Subramania Vangalaji to start your talk.
Yeah. Thank you, Rishi, and I really appreciate, and I'm very glad that I think uh, I'm pleased and I'm honored actually to be here as a guest speaker on this platform. I understand from you that most of the attendees here are actually medical doctors or similar to that, and interestingly, most of the people are also into Ayurvedic drugs. How to discover and develop Ayurvedic drugs. So natural medicine is very interestingly being pursued for a long time, and we have several drugs out in the market were inspired by herbal and Ayurvedic platforms. With that, I'll want to go and say, Bharat, are you putting up my slides? Okay, great. So I call it actually art of selecting safe and effective clinical dose. You know, I've been in this business. It's a risky business. Drug discovery and drug development, probably the most riskiest venture anybody can do, and it's not easy. I mean, I've been there many times, and you come to a level saying that, do we really understand what is a safe dose or effective clinical dose for a drug? And the decision making process is too hard. I mean, here you have a team of people, pharmacologists, medicinal chemists, toxicologists, clinicians, regulatory people, and all these people are brainstorming, even formulation development people, brainstorming to come up with a drug. And more than ever, I mean, I think 90% of the drugs that go to the clinical stage actually demise without going into the market. About less than 10% actually reach the market. Next slide, please. So, what are the two basic principles of drug development? Especially for a drug to be given to a human, it has to be safe. Safety is the most important thing. If you go to the FDA, they don't care. I mean, really, sometimes they say that, yeah, even if it is marginally efficacious, maybe we'll try it because there's no other medication to be given. But if it is too toxic, they will never allow it to be marketed. Here I'm giving different scenarios. Look at this. On the right side of the thing, I've put a balance here. In the balance, you can see if the safety and efficacy, if the safety overweighs the efficacy, then probably the risk is greater than benefit, so you don't want to. It's a no-go decision. On the left side of the uh, slide, you can see that it's a green light. Actually, efficacy is better than the safety issues. So benefit is greater than risk, and which is a clear go, green light. And you have other cases, many cases I see here in drug development are very difficult to make decisions because efficacy and safety are like balanced, which means they're very narrow therapeutic drugs. So you don't know who are the patients that will be risky for this to take this drug. So for those kind of drugs, you put a yellow light and say, hey, proceed with caution. And this is the kind of approach that typically the pharma companies do. Next one, please. So, if you know the definition of poison, this was back in early, 1500s. Paracelsus said, all things are poisons and nothing is a poison. The dosage alone makes it so a thing is not a poison. Yes, he's right. I mean, actually, anything can be a poison. But he is only referring to the dosage. In a modern world as if it is today, for the last 30 years I've been in the pharmaceutical company, I don't make decisions based on the dose. I don't consider doses the way to make decisions for this toxicity or efficacy. So we revisit. I mean, there's an article published in 2016 saying that the dose concept is a very complex word, but what is the actual concentration in the blood or tissues? That is the most critical point to address. Because so today, for the last 30 years, I know we actually measured the blood concentrations to understand both safety, which is, I mean, toxicity and efficacy issues. So for a drug to be able to be defined as a uh, I mean, highly efficacious and poorly safe drugs, you have to have kind of a drug concentration dependent analysis. So bioanalysis, the LCMS techniques, which are also highly regulated process by the FDA, we utilize to understand the drug concentrations and their kinetics, etc., to uh, understand the efficacy and safety issues. Next one, please. So, I mean, as Paracelsus said, anything can be a poison. Of course, it is. For example, you take glucose, right? Glucose is very important for us. It's an excellent nutrient for us because without which we cannot get energy. It actually provides ATP, the energy units in the mitochondrial oxidative phosphorylation for us, and that's how we survive every day. So without glucose, we cannot survive. But if this glucose starts accumulating in the blood and tissues, and you have it acylates all the proteins, anything in the vicinity, you have diabetes, diabetic complications, many people. And in fact, India is considered number one capital of diabetes in the world. So glucose is a natural molecule, but it is toxic depending on the concentrations. Same thing with cholesterol. Cholesterol is also an essential nutrient, makes the cell walls very important for the transport of the molecules across the cell membranes. But if the cholesterol is increased, especially the bad cholesterol like LDL, VLDL, you have heart disease. And you have phenylalanine. This is an amino acid, right? Some kids are born with phenylalanine hydroxylase deficiency, and these guys actually accumulate phenylalanine, and also the phenylalanine is also excreted in the urine, but it actually accumulates in the blood and brain, and it causes the mental disorders. So phenylalanine amino acid. So anything that you perturb the metabolic pathway, for even for the natural compounds, you have toxicity issues. Oxygen, without oxygen, we cannot survive. Day to day, you need to breathe. But if your mitochondria cannot handle oxygen well because of there's some uncoupling of the oxidative phosphorylation, you have oxygen spilling out oxygen radicals, which is called oxidative stress. And in fact, aging is considered to be the decoupling of the oxidative phosphorylation process, and that's why you have more and more oxygen radicals generated as we age, and that is how we age. That is one of the hypotheses for the aging process. And triglycerides, triglycerides, if you accumulate in the blood, they actually go and fool the pancreas that there's no glucose in the blood. And so you have a pseudo diabetes. So pancreas starts secreting more and more insulin. Although insulin is not the issue there. So there's actually glucose uptake problems in the liver, muscle, and adipose tissues, etc. So you have a lot of these compounds, and in fact, I wrote a couple of articles on these things. Endogenous toxins. These were all invited review articles and editorials, etc. I've been writing these things because endogenous toxins, drugs in themselves are not toxins. Sometimes you actually have cooperativity with the endogenous toxins, and that is where the science is now going after that. How do you modulate the endogenous nutrients to be toxins? Next one, please. So drug discovery process is a highly risky business. It takes $2.5 billion to make one drug into the market, and it can take as many as 12 to 15 years for one drug to come out of 10,000 to 20,000 candidate drugs that are screened. And there are essentially five parts of this. If you look at the five steps, step one is a discovery and screening, where you actually do a lot of screening against the target and we're validating the target. These are all in vitro methods, or even you sometimes use the in silico approaches to find some candidate drugs. Then the comes is step two is lead optimization. Okay, out of million compounds, you found maybe 100 hits. But out of these 100 hits, you want to know which are the real drugs. And you do the lead optimization with the combinatorial chemistry, structure-based drug design, etc. And then the next filter comes as ADMET, absorption, distribution, metabolism, excretion, toxicity studies. This is the last step before you actually go into the IND. After the IND, you actually go into the clinical trial, phase 1, phase 2, phase 3, and to be approved for NDA is the step 5. Now you have out of these either 10,000 to 20,000, only one drug that came out into the market. And that is huge productivity issue. And we're still there at the same thing. 30 years ago, I was saying 10,000 drugs to one drug. Even today, I say 10,000 drugs to candidate drugs to one drug to be approved. Next one, please. So if you look at the historical perspectives of the how many drugs have been approved by US FDA. I'm putting US FDA, I'm not looking at the Europe right now or any other country. In 1996, there were nearly 53 compounds were approved by the FDA. But if you can see that after that, the pharma companies actually have suffered productivity because it's called valley of death. The many compounds actually, many companies could not come up with new drugs. It's a poor productivity phase. There are various reasons for it. I'll come to them later. And you can see that the all-time high number of new drugs were approved was in 2018, about 59. And 2019, it came down to again 48 drugs. So you can see that the big pharma companies as well as small biotech companies are together working to improve the success rate of these drugs. In fact, many drugs actually discovered out of the small biotech companies more than the big pharma companies. And big pharma companies do in-license those small biotech companies to do the clinical trials and put it in the market at the end. Next one, please. So I kept following what are the trends, changing trends. And if you look at this, since 1960s to 1980s, the major reason of a drug that was failed in the clinic was pharmacokinetics. A lot of people do toxicology studies and without paying attention to the pharmacokinetics. And many drugs actually failed because there's not enough absorption of the drug, especially for the oral drugs. In fact, pharma companies prefer the oral drugs to any other drugs because oral is daily dose and no injections, no needles, no nothing. So they like oral drugs every day, single dose is the way big pharma works. So you can see that almost 40% drugs that enter into the clinical phase 1 actually die due to the poor pharmacokinetics. And also the rest of them which survived, in fact, 30% of them fail due to lack of efficacy. And also there are about 10% adverse effects in man, in humans, in fact, and other reasons are also there, commercial and manufacturing. And coming to the next step to 1990s, we fixed, because in the up to 1980s to 1990s, we fixed the pharmacokinetics problem. All the animal studies we do have pharmacokinetic component to it. And you can see that pharmacokinetics failures have come down significantly, but toxicity failures have gone up. So you're shifting from one side of failure to the toxicity. The reason because you have more drug in the body. Basically, that's the reason it's becoming toxic. And efficacy is really not altered. Efficacy is still at 30% failures. So toxicity has become the major concern in 1990s. Again, lots of efforts were done. People have done toxicogenomics and in vitro models and all kinds of models. Yeah, safety attritions have been reduced. If you look at the third thing in 2000s, you have reduced the safety from nearly 40% to uh, 20%, but efficacy failures have gone up in the phase 3, especially in the last. So right now, phase 3 efficacy failures are about 90%. Really hard. If you take a molecule up to the phase 3, you have already spent nearly $1.5 billion, and now you go to the phase 3 trials and you're failing the molecule, and that's a big blow to the pharma companies. All their budgets are gone because of these 90% failures in phase 3 stage. Next one, please. So how do we do the safety assessment? Typically, this is a regulated process. Although you do early screening, trying to understand the genetic toxicology, safety pharmacology, cytotoxicity, SAR alerts, photo and UV toxicity, etc. You do a lot of screening to select the lead candidates. And the lead candidates then go into the IND enabling programs. There you have to do the GLP toxicology mandated by the FDA. Whether you do genetic toxicology, safety pharmacology, acute toxicity. For IND, depending on the type of the molecule, you can do a 2-week toxicity study or a 4-week toxicity study. Typically, the FDA insists on one small animal like a rodent, rat, or mouse, or a non-rodent like a monkey, a dog. And in recently, people are also combining biomarkers like toxicogenomics, metabolomics, etc., to get a better understanding in the animal studies. But that is still not a regulatory process. The regulatory process is to understand what is the toxicity uh, that you're seeing in the animals, number one. And once the IND is filed, your compound is safe and you have very good molecule to go into the clinical trials, then you have to still continue in the non-clinical toxicology studies. Majority of the studies go up to 1 year, depending on compound. And you still need to do rodent plus non-rodent. And you also have to add reproductive toxicology and environmental safety and occupational exposure studies, etc. Then you go to the NDA filing. Based on all these data, if the FDA is satisfied, sometimes they say that, okay, we'll approve it, but we're still not concerned. There may be a non-genotoxic carcinogen because of the mechanism that structural alert is coming. So we want you to do post-marketing carcinogenicity assays. Then you also want to do pediatric. You want to go to the kid. Whatever is approved for the adult dose is not approved for the kids. Period. A lot of doctors try to. I mean, I'm not blaming doctors. A lot of doctors trying to give the same dose to the kids as well. That is not allowed. We have to do animal juvenile toxicology studies to ensure that doses can be adjusted for the kids. In fact, there is phylogenetic programs that say that the drug metabolism, drug transport are actually different in a developing young adult up to till 13 years. So you've got to be extremely careful doing the pediatric dosing with the adult uh, prescribed dose. And you also need to, depending on the molecule and other issues that you may see during your drug development toxicology studies, you may have to do the local tolerance, immunotoxicity, and phototoxicity studies. This is the entire package that is required for the FDA, but you can negotiate with them depending on the urgency and because there's no other molecule in the market and this is urgently needed for the terminally ill patients. So you can negotiate with the FDA to not do some of these studies and fast track the molecules. Next one, please. So As I said, pharmacokinetics is an integral part of the toxicology, even clinical studies. So for this, we do a lot of studies. For example, if the compound is not soluble, it is highly fat soluble, not water soluble, it will not absorb through the intestinal tract, especially for the oral drugs. And the drug also has to permeate the cell membranes. If it is not permeable to the cell membranes, then you don't have a drug. And of course, log D, protein binding. Protein binding, if some drugs have 99%, they're very, very, especially warfarin, it's got 99.9% protein binding. It's considered very narrow therapeutic drug, even if you actually decrease the 99% protein binding to 98% protein binding, it could actually cause bleeding in patients. And you have secondary screens that we do to understand what is the permeability, what is the cytochrome P50 enzyme that metabolize the drugs, and are there any potential for drug-drug interactions, especially old people, about 55 plus people. They may be taking multiple medications because they have multiple complications. They have arthritis, they have diabetes, they have some psychological problems. So they're taking more and more drugs. In fact, if somebody takes more than 4 drugs at a time for continuously, and they may have fatal drug-drug interaction issues, and some people die of cardiac arrhythmias or liver toxicity, other issues. So we do all these studies as well to ensure that we don't have any liabilities. Then you do the animal studies. It's a must. We take at least 4 animal species. So minimum mouse, rat, dog, and monkey. We do the pharmacokinetics and bioavailability studies. And we also try to understand the formulations, because in drug discovery, when you do a pharmacokinetic study, you'll say, oh yeah, the drug discovery chemist comes and says to me, oh, I have 80% bioavailability. But when you actually go into the regulatory drug development, you have to have a single polymorph, not multiple polymorph. So the stable polymorphs are usually very unstable in the solution. And so what happens is, multiple polymorphs are unstable. So it actually the drug conforms to the stable polymorph, which is usually very insoluble. And so whatever you get the pharmacokinetic bioavailability data in the drug discovery phase doesn't meet the requirement for the regulatory filings, because your bioavailability will drop down to 15%. And that is going to be a very hard sell to the FDA. Unless your drug is highly potent, that picomolar concentrations can bind and be efficacious. So you can look at your efficacy data and see if your 15% is okay for you. But typically, 15% is a cutoff for me bioavailability. I don't take any compound into these. Now you have 4 species of animals that we scale it up to understand what will be the human concentrations of the drug. What is the PK going to be? So you do a PK/PD modeling on these animal studies and try to come up with a approximate. These are all empirical models. These are not really very predictive, but empirical models, 30 to 40% predictive models. But we still do it, telemetric scaling, to be able to do it. Then you go to the human, and then you repeat and see you're validating whatever you predicted in the animals is going to be showing up in the humans as bioavailability or drug-drug interaction or pop PKPD, because pop PKPD is very essential, because in humans the variability is much higher than in animals. And in fact, another one is the metabolite identification has to be done very early on, because a lot of drugs, the toxicities are due to the metabolites. And in some cases, the metabolites are the actually efficacious compounds. Not the parent comp. So parent becomes a prodrug and metabolite is the actual drug. So you need to understand what is the relationship between the metabolite and the parent drug pharmacokinetics. So next slide, please. Hello? Yeah. So when we are doing the screening, we'll take all these compounds and say, and try to put them in order. Yes or no, like green, yellow, and red. Red means no go, green means go, yellow means proceed with caution, as I said. So the more greens, more preferable to go into the IND enabling studies. More reds means no go, you don't want to go to the IND enabling studies. Some greens with some yellows may be manageable, maybe very one or two reds may be manageable if you're okay with it and take it forward. I mean, you have to do a rational justification with a couple of reds to be able to go into the IND enabling studies. Next one, please. So there are lots of major guidance documents to be filed for filing IND. IND filing, FDA has many guidance documents, non-clinical safety studies for conduct of human clinical trials, preclinical safety evaluation of biotechnology-derived pharmaceuticals, genotoxicity testing, safety pharmacology studies, in vitro drug-drug interaction studies, especially with cytochromes P450 and drug transporter mediated. These are all regulatory studies must be done for the FDA. Physiologically based pharmacokinetic analysis. And this is also catching up now because a lot of people want to do this. So exposure response relationships, we need to understand what is the exposure in each patient and how does it relate to the efficacy or safety. And of course, the toxicokinetics in animals has to be able to predict the toxicity in the humans. And of course, again, FDA insists on safety. If you have metabolites in animals, but you have a different metabolite in the human, or the metabolite is with higher exposure in the humans, FDA will ask you to do the safety and toxicology studies all over again for the metabolite. Okay, so the metabolites are very, very crucially to be understood before you go into the phase 1 trials. Next one, please. So this is the guidance for industry, just putting up how they look on the FDA. If you go to the FDA.gov, you can see all these guidance documents. If you can click it, and you'll get it. Next one, please. So major objective, as I said, is animal toxicology study is not just a what is that? Checkbox studies. These are not checkbox studies. There's a lot of science goes into it. The major objective is how to come up with a safe first in human dose. That is the reason you're doing all these animal toxicology studies. Otherwise, there's no need to do this. If it is a checkbox, then there's no need to do it. So, pharmacology studies in relevant animal models and in vitro models, efficacy exposures must be obtained. General acute and repeat dose studies in 2 animal species, as I said, 2 animal species are required. Toxicity exposures must be determined. Genotox studies, safety pharmacology studies, especially safety pharmacology, cardiovascular safety pharmacology is essential. Toxicokinetic data must be obtained. Pharmacokinetic studies in animals, determination of bioavailability and dose linearity. Dose linearity is very essential. In vitro human drug metabolism studies, including human, because we want to understand the metabolism differences from the animals to the humans, whether we could plug these also into the scaling factors, drug metabolism enzymes, drug transporters, and then of course drug-drug interaction studies. Many drugs have been withdrawn from the market after they are marketed because many patients died because of the drug-drug interactions. Identification of drug metabolites using in vitro systems and also in animals. Determination of maximum tolerated dose, no observed adverse effect level, interspecies scaling of pharmacokinetics, PK/PD modeling are very essential tools to come up with the safe first in human dose. And pre-formulation studies are also very important because you want to know which formulation you can take into the clinical trials. Sometimes you want to do a solid dosage. You don't want to go with a liquid formulation. Then you need to come up with a pre-formulation program to be able to come up with a solid dosage formulation. There are lots of other special studies you need to do. phototoxicity, accidental overdose, reprotox studies as recommended by the agencies. Even before IND, sometimes they may request you to do this, depending on the chemical structure. Or if you're going into the pregnant women, you're not allowed to. So many people don't want to go to the pregnant women in the for the reprotox teratogenic studies. But if there's a case to be made, then you have to show the FDA relevance of this. So it doesn't cause any reprotox. Next one. Skip this one and next one, please. So I'm giving you here just a view of hydroxychloroquine. I'm actually writing an article on hydroxychloroquine controversy. So I prepared this slide for it, but anyway, I'm putting it up here. So the hydroxychloroquine is an oral drug. It goes to the intestine. It's in the intestine. Some of it actually gets not goes into the portal vein because there's a P-glycoprotein drug transporter that actually is an efflux transporter, prevents it from entering. And then once the through whatever the drug that goes to the portal vein, uh, then reaches the liver, and HCQ can be metabolized there. And a lot of drug metabolism enzymes like P450s, EDP gluconyl transferases, and other enzymes. And then it goes into the tissue distribution mode and finally gets excreted. And you can see that other drugs, herbs, cytokines, etc., can inhibit the drug metabolism enzymes. And also cytokines actually do downregulation also sometimes of the drug metabolism enzymes. Some other drugs and herbs can also induce the cytochrome P450 enzymes, so the metabolism is accelerated. So you have to understand, especially the other organs, heart, because the heart cardio toxic QT prolongation is a major issue these days. FDA won't take your drug into the clinical trials unless you show that it doesn't affect the heart. So you have efflux transporters, uptake transporters, drug metabolism enzymes. They all play together role. And some of the drug that goes into the liver, either as a metabolites or the parent drug, can be gone to the bile. Depends on the molecular weight. And the bile, either it can be excreted or some of it partially re-enter through an enterohepatic recirculation into the portal vein. So this is a complex pathway of the drug disposition of orally administered drugs. Next one, please. So I told you we need to know the bioavailability of the drug, right? Bioavailability is simply how you determine that you give an IV dose, say 1 milligram per kilogram dose, and you give an oral dose, 10 milligram per kilogram, and you see the plasma drug concentrations. And you can see the plasma drug concentrations. Oral, typically the ideal half-life for any pharmaceutical drug candidate, ideal acceptable is 8 hours half-life. Anything beyond that, you got to be careful because it may accumulate on multiple dosing. The typical daily dosing requires 8 hours half-life. And AUC oral, you can see that you can say, okay, because you're directly injecting the drug into the blood, you call it 100% bioavailable. Now you calculate the AUC for oral and you come up with say that what is the percent bioavailability for oral administration. You can also do that for skin administration. You can do the buccal administration against the IV injected material to come up with the actual bioavailability. I've just put up here one thing, and you can see that there's a lot of variability in clearance. Especially you look at 1.25 or in one patient is 0.58, the concentration clearance goes always to 0.592, and you can see there's a lot of variability here. 0.241, so 0.138. So you have too much variability in the form. But if you put the PopPK models, you can say that, okay, these are can be explained by gender differences. Males versus females, there's a huge difference. You can say, oh, okay, males are handling the clearance is higher. Sorry, clearance is higher for females compared to the males. And so the drug disposition in females is different. I tell you that females have also hormones, higher levels of hormones that may actually affect the drug metabolism as well. So you need to be careful when you do gender differences as well. Next one, please. So there are two pharmacokinetic bases, a linear pharmacokinetics and non-linear pharmacokinetics. You can see the graph there. I'm putting here just a empirical. I mean, I just pulled the numbers out of my hat. They're not from any paper or anything. You have dose escalation from 50 milligrams to 1000 milligrams per kilogram. And if you can see, if it is a linear, the AUC by dose should be a linear concept. But you have two different cases here, nonlinear PK. In one, actually exposures are increasing with the dose, higher, very high. But that means it's accumulating in the body. Whereas in the other side, actually exposures are not altered at all. It's kind of flat. Whether you increase the dose how to 1000 milligram per kilogram. So this is where dose doesn't matter. The concentrations matter. This is the most important factor of the toxicology studies is when you're designing, you need to do the dose linearity and understand what is your drug disposition properties. If you go and just do the toxicology studies with 50 to 1000 milligram per kilogram, if the drug is not showing the linearity of the exposures, you're really wasting your drug and your time in doing toxicology study. Okay, so basically there are two reasons why these things. Inhibition of drug metabolism or drug transport by the drug itself, causing the increase in the nonlinear PK1 model, or poor drug absorption in the gastrointestinal tract, or increased or induced metabolism in liver is nullifying the dose response. So these are the two reasons why you need to know the metabolism as well. Next one, please. So here, how do we do the allometric scaling of pharmacokinetic drug clearance and estimating the human dose? Human PK actually. So you take the 4 species. As I say that, typically what we do is in pharma industry. I took this from this article, Snare Netra. I do have my own publications, but anyway, this came out very easy, handy to do it. Typically we do mouse, rat, dog, and monkeys. If you look at the body weights, mouse is only 20 grams, rats is 200 grams, monkeys are about 3 kilograms, and dogs are 10 kilograms. And you extrapolate for the body weight for a 70 kilograms human. So if the pharmacokinetics are extrapolatable to the 70 kilogram humans, that means this drug doesn't have any issues in terms of the metabolism, drug transport, or any other issues that confound this. So these are all 4 examples there, and you can see that in all these cases they're showing a linear response against the body weight to the to predict the human PK for the these 4 drugs. Next one, please. So conversion factors. So how do you convert the animal doses to the human equivalent doses, right? This is there in the literature. You can find anywhere. Lots of even FDA or ICH guidance documents have it. I only picked the 5 species that are relevant for the pharmaceutical industry. If you want to estimate for the human 70 kilogram and you have mouse, the body weights, and if you actually look at to convert the human equivalent dose, you can either divide animal dose by these numbers or multiply animal dose by the other numbers. That's a simple way of converting the animal doses to the human doses. Next one, please. So how do you now come up with the maximum recommended safety dose for going into the first in human clinical trials? Typically in toxicology studies, you have a no observed adverse effect level dose. And that is very critical, which means there's no adverse effect. And of course, you have also the toxicokinetic data from those studies. So if you take rat, it is 50 milligram per kilogram. Dog, it's very sensitive species. You can see 15 milligram per kilogram. In monkeys, again, 50 milligram per kilogram. So in this toxicology studies, you're showing that dog is the most sensitive tox species. And according to the FDA, you have to actually take the most sensitive species, not the most non-sensitive species in terms of the toxicity. Then you calculate from the previous slide I showed the multiplying factors. You can calculate what are the human equivalent doses. 8 milligram per kilogram, 8 milligram per kilogram, and 16 milligram per kilogram will be the human equivalent doses. So if you look at that from there, according to the FDA, even if you take the most sensitive species like dog, you still need to, if it is 8 milligram per kilogram, then you have to divide by 10, factor of 10, and you can only start the dose at 0.8 milligram per kilogram. This is what FDA is a very conservative first in human dose approach. Here they're not interested in the efficacy. The first in human dose should be safe. Once you go and you get your pharmacokinetic data from that first in human dose and you validate against what you predicted from the animal PK, and if you are on the same line that your animal the pharmacokinetic data, now you have no observed adverse effect level toxicology concentrations, then you can escalate the doses as much as possible until you reach the no observed adverse effect level toxicokinetic concentration. You cannot go beyond that. It's impossible because FDA will not escalate your doses. And if your efficacy is not evident by the dose, that means you lost your game. That's it. You cannot go back and ask the FDA that I want to escalate the dose beyond these concentrations that I have in the past. So for a 70 kilogram human, a dose of 56 milligrams can be the FIH dose in these calculations. And I'm telling you, this is only 30 to 40% times predicted. Not all 100%, you can never predict it. So you need more scaling factors, need to be incorporated for a more accurate prediction of the human first in safe dose. Because many times I've seen, I predict the human PK and I go and hey, either way off, sometimes very way off. Either it is very 10 times lower than the predicted or 10 times higher than the predicted. And you really come back to the square one and blackboard and what do I do there? So these are very essential studies to be able to convince the FDA that you have the safe dose and that could be an efficacious dose as well in the humans. Next one, please. So what are the factors that create this poor prediction of the human pharmacokinetics through the allometric scaling approaches that I just described? Target polymorphisms. Many targets are polymorphic. In fact, but that doesn't have to do anything with pharmacokinetics, but the drugs don't work if they're inactivating polymorphism of the gene, so the target itself is not going to work. And then you have drug metabolism enzymes polymorphisms, drug transporter polymorphisms, inflammatory mediators. For example, you're talking about cytokine storms in COVID-19. All these cytokines actually can downregulate the cytochromes P50. So your drug metabolism of the hydroxychloroquine could be very different from a patient with lupus or rheumatoid arthritis or a malaria patient. So these have to be taken into consideration because COVID patients are also taking many other drugs for other diseases because lots of comorbidities are there. And poor specificity and selectivity, drug-drug or drug-herbal interactions. This is also very important because sometimes a patient is taking 2 drugs or 3 drugs. They could be metabolized with the same enzyme or inhibiting one enzyme or something or inducing another enzyme. So you have the drug-drug interaction. So basically these are the reasons why sometimes animals are poorly translated to the humans. Next one. So cytochromes P450, these are the most well-studied enzymes in the last 40, 50 years. It was discovered in 1950s, I think early 1950s. Now if you look at the phylogenetic order, I mean, if you look at the mouse, rat, dog, monkey, and human, they're all different. They're all different. Mouse enzyme, for example, I'll take CYP3A, right? There are 3A, 11, 13, 16, 25, 8, 41, and 40, multiple P450s. Whereas in humans, 3A4, 5, 7, and 43 so far identified. And 3A4 is the major metabolism in the adults, metabolizing 3A4 in the enzymes. If you take 3A7, that is actually a placental enzyme. So if you go into the neonatal or some other subjects, then you cannot use the 304 data. And in fact, cytochrome P453A isoforms metabolize 50% of the drugs on the market today. And the second enzyme that comes is that 2D6. 2D6 is a cytochrome P52D6, metabolizes about 30% of the known drugs. But if you look at the cytochrome P453A content and 2D6 content, in the bottom table, 2D6 is only 2.5% of the total P450 levels in the liver, whereas 3A4 is about 60% of the total P450. But the variability, if you look at 2D6 expression in the human livers, there's 1,000-fold variability in the populations. So one person versus another person, there's a huge difference. Especially if a drug is metabolized by 2D6, you have to consider the pharmacogenetics disposition and looking at the genetic differences in the populations. You don't have to do for 3 or 4 the same thing. Next one, please. So, actually, I thought I had another slide in between. But anyway, so drug metabolism and its consequences. That's okay. Yeah, fine. Leave it there. Drugs are metabolized in the liver to phase 1 metabolites, and these phase 1 metabolites can go to the phase 2 metabolites and be detoxified and excreted. Very rarely phase 2 metabolites are toxic, except for the anti-cancer drug one. I forget. There's a glucuronide metabolite. It's very toxic. Anyway, the other consequences is that many of these drugs are also produce reactive free radical and electrophilic metabolites. These doesn't float in the blood plasma or tissues. They are conjugated to they covalently bind to the proteins, DNA, and all the other macromolecules in the cell. So you don't see them directly. If you really want to detect these metabolites, you need to throw in a nucleophile like glutathione and identify a drug adduct with the glutathione or something. So you have a covalent binding to the protein that causes actually hapten hypothesis for immunotoxicity, right? And this is exactly what happens is many of these drugs actually do cause reactive metabolite producing drugs do cause immunotoxicity. And I see many companies in the US, big pharma companies, they never do the reactive metabolite screening in the early development until when they see a real problems in the clinic or somewhere else. The exception is Merck. Merck does, in the screening process itself, they'll screen for the reactive metabolites thoroughly to make sure that they don't have these liabilities in phase 3 trials or after post-marketing. Next one. So this is actually intraethnic and intraethnic variations of CYP2D6 genetic polymorphisms. This has more than 100 alleles, CYP2D6. And you can see, I don't want to go through this entire table, but I'm just telling you that even Indian populations are also characterized. There's the major isoform is the polymorph is star4, which is actually also different in terms of the percentage. Maybe it also, across the ethnic species, that is the major one that actually metabolizes the drug. But you have non-functional alleles. If you can look at the normal function versus decreased function versus no function. So there are lots of non-functional alleles, which means, so the Caucasians, it is expected that 10% of Caucasians don't metabolize if they're CYP2D6 deficient, because what happens, CYP2D6 is deficient in 10% of the Caucasians. And if you go to the German Caucasians, they may have about 5 to 7 copies of the CYP2D6, though they metabolize all the drug instantaneously, so the drug won't work. Whereas in the Caucasians, that there's no CYP2D6, the drug is too high concentration, it becomes toxic. So these are the reasons you need to understand the genetic polymorphisms, especially with regard to the 2D6 and maybe 2C8. These are the two isoforms. Next one, please. So the transporters, drug transporters, these are also being, now FDA has actually issued a guidance document, it was published in January, that they're also giving importance to understand the transporters. For example, if you look at the intestinal epithelial cells, right? So intestinal lumen, you have transporters. Majority of these are actually efflux transporters. They don't let the drug in to the systemic circulation, portal vein. The reason, because it's adaptive response. It doesn't recognize it. It considers the drug as a foreign molecule and it throws it out. I don't want you there. It's a guard, basically, Gurkha. And whatever the drug that is absorbed goes into the portal vein, and portal vein, it goes into the liver, hepatocytes. You can see there are several transporters there at the hepatocyte. A lot of people do the in vitro hepatocyte studies, and many times they don't see a drug getting metabolized. But the same drug is actually metabolized extensively in the humans. The problem is, when you're taking the in vitro in hepatocytes, many of these transporters are downregulated because of the stress when you're isolating them. So you don't get some of these drugs need the transporters to get inside the hepatocytes. They can't get in vitro models. So you need to be, some of these studies are in vitro studies are flawed because they don't understand this biology of these transporters very well. And so if you look at, there are also other, to go to the blood, there are lots of other transporters also, efflux as well as, and some of them also go into the biliary transport. So you also have brain. Brain also needs transporters to get inside. If these transporters are not expressed, the drugs cannot penetrate the blood-brain barrier. So kidney is another organ, because kidney is the excretory organ. All the drugs, majority of them are excreted through the kidneys, and these kidney transporters also regulate the excretion pathways of these drugs. Next one, please. So the other point is that induction. Many drugs can actually activate a nuclear receptor, especially CYP3A4. This is an example for CYP3A4, pregnant X receptor, and it forms an heterodimer with RXR when a drug binds to these ligands. Not only drugs, you can also see some nutraceuticals like hyperforin, St. John's wort, kava, they also bind. And what they do is they actually bind to the PRE element on the gene, regulatory element of the CYP3A4 gene, and transcriptionally activate it, and you can produce all these even transporters, CYP3A4, CYP2D6, CYP2B6, all these enzymes are controlled by this PXR, RXR element. So this is very interesting. And also there are grapefruit. It's very interesting that in 1990s, people were taking some drugs, and they were actually taking the grapefruit juice, and they had sudden cardiac arrhythmias and died. Grapefruit juice actually has a compound called bergamottin, and that bergamottin is a very potent inhibitor of the drug transporters and CYP3A4. And with that, we realized later on, and that now, instead of removing the grapefruit juice from the market, you cannot remove the grapefruit juice from the market. So those drugs were withdrawn from the market, because you cannot ask people to not to drink grapefruit juice. So these are the things that need to be understood, that even nutraceuticals actually interact with the drugs, and that is sometimes very dangerous as well. Next one. So the other I already mentioned that inflammatory conditions or infectious agents do cause inflammation, right? And inflammation, your drug metabolism is different. So in disease conditions from the healthy patients, the drug metabolism is going to be different. All the phase 1 trials are done in healthy patients, majority of them. But if you go to phase 2, phase 3 trials, you're doing in the diseased patients. Your pharmacokinetics is going to be different. That's why phase 2 and phase 3 trials fail, although phase 1 trials are, encouraging, right? So if you take an unhealthy patient, uh, they have inflammatory disease already, and there are lots of pro-inflammatory cytokines. They downregulate the CYP enzymes, and there's no CYP enzymes in the liver. So the drug metabolism and clearance is not there. So the drugs concentrations go way up to toxic level, especially with hydroxychloroquine. This is one of the things that doctors haven't realized, that why hydroxychloroquine is not being approved by the FDA. The increased concentration of this drug can actually cause fatal cardiac arrhythmias. And in fact, hydroxychloroquine plus azithromycin, together the combination, even azithromycin causes arrhythmias, and together it could be more fatal. That's why FDA is not listening to the doctors and saying that, okay, hydroxychloroquine, we don't want you to dose patients. Besides the fact hydroxychloroquine mechanism is still not understood, I don't think it's actually efficacious against the COVID-19 as well. If you take a healthy patient, and basically patients that don't have any disease, but they have infection and inflammation, they have also secrete the pro-inflammatory cytokines. Here, downregulation of cytochrome P50 enzymes. Actually, the drug is already metabolized extensively, but it's actually they're going to be metabolizing more drug because of the drug relation of the CYP enzyme is going to change some of these enzymes. Next one, please. So many drugs, as I said, were withdrawn from the market after they were marketed. For example, troglodyone. It's an anti-diabetic drug. In 1999, I think Warner Lambert, now Pfizer, marketed this drug. Fantastic PPAR gamma, I think PPAR gamma antagonist, working very well for the patients. Within 1 year, there are about 3,000 patients died of drug-induced liver injury. And it is attributed to the toxic reactive metabolites. This is very surprising because animals don't show this idiosyncratic toxicity. And this is not dose-dependent toxicity in humans. And this is where cannot be predicted from the animal models. And you can see many other compounds that have been marketed but withdrawn over the years. Many of them actually do produce reactive metabolites that actually cause immunotoxicity. Majority of them do cause immunotoxicities, and some of them do, like trophoxacin, cause mitochondrial dysfunction. They actually uncouple the oxidative phosphorylation. Next one, please. But still, there are some drugs. The FDA is still reviewing that the data very carefully. And amoxicillin clobelinate. I mean, in fact, any doctor you go to immediately in India, this is the very potent antibiotic, amoxicillin clobelinate. You should take it together for your infection. It causes cholestasis, which is like a jaundice, and mixed hepatocellular necrosis. And it causes immunoallergic reactions. So FDA is actually carefully reviewing the data now to see what to do with the unusual cases of these kind of cholestasis. And so you have several cases of these drugs are still in the market. Carbamazepine is an interesting drug, and it is also metabolized by one enzyme, which forms an epoxide toxic metabolite. So carbamazepine, in very rare circumstances, FDA recommends do the actual analysis of the carbamazepine and its metabolite in the plasma for the patients. It's required. They say that we cannot do it. The same thing I tell people, the doctors also, for hydroxychloroquine. If you want to dose the hydroxychloroquine to the patients, please do the pharmacokinetic analysis along with and also at the same time you do the ECGs if you think this is the only medicine that can save that patient. Okay, next one, please. So same thing, some drugs withdrawn from the market due to cardiac toxicities. There are arrhythmias, there are cardiac valve dysfunctions or other ischemia functions. There are lots of drugs also. So these are the major two toxicities: liver and cardiac toxicity has to be paid attention very early on during the early drug discovery potential for these toxicities. And the third one is the kidney toxicity. Okay, next one. I just 1 second, go back please. I listed the Posicor. Posicor was a calcium channel blocker. It was also released in 1998, I think, by Roche at the time. Wonderful drug. It was making $2 billion within 1 year. But the problem was about 5,000 to 6,000 patients died because they were actually on many other drugs, because most of these calcium channel blocker patients also have other drugs. And at least 16 other drugs that they listed, this drug could interact with. And so that is the reason explained for the fatal drug interactions for the Posicor. And Posicor, I think FDA, sorry, not FDA, Warner Lambert paid about $4 to $5 billion in compensations to the patients that died for the troglodyone. Same thing, about $2 billion for the Posicor. Roche paid for the patients' families. So it's a risky business. You spend $2 billion to make a drug into the market, and now that drug causes toxicities that you didn't know didn't know that would cause it, and now you have to pay from your pocket $5 billion. You're bankrupt totally, and that's why many companies go out of the bank market and say, "Hey, we are for sale." Like that's how Pfizer is buying all these companies. I was working for Wyeth. Wyeth also had problems with cisapride and other drugs. It was taken away. Not cisapride, something, dexphenphoramine, Redux, and all kinds of other drugs. So they immediately said, sale board, and Pfizer bought it. So this is one of the problems, risky business. We cannot sustain for too long in drug development business. Next one. So to be honest, I didn't do much of dermal drug discovery or something. We know there's a lot of cosmetics are being looked at the efficacy for the dermal. It's not efficacy, but cosmetic point of view. But anyway, I mean, many issues are there for the drug penetration. The one-way drug absorption is good because from dermal, because dermal cytochrome P450 is here, about less than 20% of the liver concentrations are there. So metabolism is not an issue. We don't know yet the drug transporters how much role they play, but the advantage is that because of there are several layers, and the upper layer is not enough moisture, but in the bottom layer of the dermis, you have about 70% moisture. So drug spilling into the systemic circulation is very low. We were trying to do a patch because opioids, especially buprenorphine, were very, very toxic, especially fatal at the CNS toxic effects, liver toxic effects, and other things, orally administered. So if you actually administered with a patch, there's a sustained very low concentrations that don't go into the CNS for the drug to be abused. So these are all the ways you can use the dermal absorption models to be able to stunt the drug absorption and sustainably relieve. You don't need sustained release formulation by oral mechanism. You can actually get a sustained release by just putting a patch or some cream on the skin. Okay, next one, please. So there are lots of skin toxicities issues by drugs. These are even for systemic drugs. Many of them, and I'm not going to go into the details. This is self-explanatory. You have contact dermatitis, photosensitivity. Photosensitivity is very important because many orally absorbed drugs, when they reach the skin, if the person goes into the sunlight, and it can be phototoxically activated to a reactive toxic metabolite, and that can cause the immunotoxicity in the skin, and you have skin lesions over and over, hypersensitivity reactions. So it's we got to do the photosensitivity reactions early on in drug discovery to be ensured that your drugs don't carry the liability of the phototoxicities. Next one. So what are the evolving complementary in vitro models? As I said, animal models alone cannot be helpful. So we've been working for the last, I think since 1980s, I've been working on using human in vitro models primarily. You can in the 1980s we used to simply take the liver, grind it up, put up the subcellular fractionation, and characterize the enzyme specificity and sensitivity. As we moved into the 1990s, we got into the cell models, 2D cells, tissues, recombinant enzymes as well. Many of the cytochrome P450s are expressed in the animal models, transgenics, and also knockout knock-ins, but also in the E. coli, yeast, all these heterologous expression systems. We use them to study mechanisms in vitro for these cytokines before. As we moved on into 2000s, slowly 3D cells on tissues and regenerative stem cells have come up. Now people are looking at more of cell-cell communication, multiple target identification, pharmacology, and toxicology. For example, liver is not a homogeneous organ. It has got 5 different cell types. Parenchymal cells make 50% of it, whereas you have fat cells, endothelial cells, Kupffer cells, macrophages, which are actually also endogenous macrophages. They can be inflammatory cells. So all these things you cannot predict just by using only hepatocytes. For example, if you take the classic example of paracetamol, paracetamol is metabolized in the liver to a quinolamine reactive metabolite that binds to some critical proteins and damages and causes necrosis in the liver cell. That damage is not enough to cause a liver necrosis, but that attracts the inflammatory neutrophils from the systemic circulation, and they come and start damaging more of the liver. That is why paracetamol at high doses becomes really, really damaging because of the not only, so you cannot modulate that just using only one cell type of hepatocytes. You do need inflammatory cells in conjunction with the hepatocytes to predict what happens by a drug in the liver when you have 2 or 3 different. Another issue is that there are some drugs that are metabolized in the liver, goes to, for example, in bone marrow, further get metabolized to reactive metabolites. So if I don't have a reactive metabolite in the liver, it doesn't mean that reactive metabolites are not produced in other tissues. So we need to be able to cognizant of all these multiple issues. So you do need cell-cell communication, organ-to-organ communication. Now microfluidics with multiple organ interactions, pleiotropic effects, clinically relevant models are coming up. And this is the next generation technology even we at Reagene Biosciences are working on to be able to better predict the human safety and efficacy for drugs. And these are the models we are trying to patent and look at that. So these are also cost-saving models because in clinical trials are very expensive, and if you go to the clinical trials and you fail them, you already burnt $1 billion. So these models can help you to save money, not fail late, fail early. You can actually fail earlier than too late. Next one. So Dawn of the Zombies. When I was talking about the microfluidic platforms, 2003 to 2006, I actually was J&J built the prototype model for the microfluidic organ on a chip model. And there was a freelance writer at the time came and interviewed me, new scientist, actually credited me for developing these prototype models. Next one, please. And she actually, the freelance writer mentioned that Subramaniam Wangala, regional head of drug development for the US East Coast of Johnson & Johnson, was so impressed when he heard about these models, human on a chip concept about a year ago that he joined forces with Hurel. Wangala hopes new approaches like the human on a chip will give drug discovery a much needed boost. And this is where it comes from to me that it's become a passion for me to also build these models to be able to uh, reduce the drug discovery timelines, costs, and improve the predictive values and improve the drug success, success of drug discovery models. Next one, please. So this is what you want to do is you can actually use the 3D models, and this is the logo that we have for the our company, Reagent Biosciences. Next one, please. So I think if you ask me what is the future, now what we are doing is with these in vitro 3D models, we're actually looking at the network pharmacology, using systems biology, networks, also network toxicology and network pharmacology, along with drug discovery and development applications, and also utilize biomarkers. So what you can do is with all these systems, when you put into 3D model in an organ to organ communication model, then you actually have a better handle on the human pharmacology and human toxicology, how that works in relation to the humans. And this is what our vision is for Reagent Biosciences. And we hope to be successful one day to come up with these models for regulatory to accept these models as alternatives to animal models. Thank you very much.
Thank you, sir. It was really an enlightening session from you regarding the regulatories as well as toxicity, and how you have really given us a clear insight on how important this toxicological investigation prior we go for classical drug discovery process at different stages. And now I request panel members to unmute yourself if you have any questions and get the answers from the speaker. Thank you.
Bharat, you can start with the questions which are already there in the chat box.
Okay, so hello, sir. I would like to take the questions from audience. We have a question from audience. Mr. Dev Anand has a question like, does non-animal data accepted by FDA, is it is mandatory to conduct a in vivo uh, toxicity and efficacious data prior we go for IND registration?
Absolutely must. FDA will not entertain without animal data as yet, because there's no other model yet as yet for the, because at least with the animal models, you with the empirical scaling models, you can enter into the first in human safety studies with a conservative safety dose. You cannot do that with the available in vitro models as yet. So the only place where the regulatory agencies said you don't need animals is the cosmetics at the moment. Okay. Uh, and slowly may extend to the nutraceuticals. And nutraceuticals also, you can avoid some of the animal studies. You can show if it is already there, people are eating diet every day and everything. And so you can avoid a lot of these studies for nutraceuticals. But for drugs that are synthetic drugs, there's no way you can avoid these animal studies at the moment. But they are entertaining, and they're actually working with some companies like Emulate and other companies and trying to validate these in vitro models. And this is what I also want DCGI to do in India, that they should be active like FDA to come up with these kind of technology platforms to help out. I mean, in the last year with ICMR, in fact, there was a paper out, Need for Alternatives to Animals, for not only drugs, but for many other platforms. And in fact, 80% of that document was written by me. And ICMR published it in IJMR. Of course, I was one of the expert committee members for ICMR. But it has already there. DCGI should now encourage Indian scientists to build these more and more of these models to be clinically relevant models, not non-clinically relevant models, to be able to improve the drug discovery. Because India has never been on the forefront of drug discovery platforms and technologies, and this is the opportunity that we need to avail and build these platforms for India.
Thank you, sir. I think the question is very well answered, and I would like to take the next question from the audience. The next question is from Dr. Murali. The question goes like this: considering the genomic difference between humans with fish or Drosophila, how to correlate the results obtained from these experiments to humans? He is requesting you to comment on this.
I mean, I'm not a Drosophila fan. Because I mean, even the animals are not able to predict the human uh, drug efficacy and toxicity. How can Drosophila predict it? So, I mean, I think they're okay, good to the research angle models, but they're not regulatory models. I cannot use them as a regulatory models. FDA will not accept them.
Thank you, sir. And I have a next question. What is your opinion for conducting early toxicity studies and PK studies on disease models to mimic clinical scenario?
So, I mean, in the clinic, the major issue is that the drug, we don't even know what its PK is, number one. Number two is that you don't know what kind of safety issues are there. Yeah, you're extrapolating from the animals and going to the clinic. Directly going into the patients is unethical because the patients may be already on other drugs, and you cannot pull them out of those drugs to do research on your new drug, which it may not even work. So it's ethically not considered to be, except in some cancer studies. If there's no other drug for the patient, FDA may allow you to go with a new drug because there's no other drug available for the patients to survive. In such cases, you can make a case to the FDA. But otherwise, you have to do the phase 1 trials in healthy patients to make sure that there are no, even in healthy patients, if there are safety issues, disease patients are going to be more safety issues. So you need to do the healthy patient studies first before going into the uh, diseased patients.
Thank you, sir. I think the question is answered in detail. And I would like to take the next question from Sachin. The question goes like this: can you please suggest some animal models that can be used to study neurological pain management using plant-based molecules?
Okay, again, I'm not a neurological expert to tell you the truth. The problem is that, I mean, I may not be answering your question exactly, but here it is, my take on it. CNS is a very tough organ. The major issue with the CNS is the blood brain barrier. And if you don't understand the blood brain barrier, in vitro animal studies or animal studies really doesn't help you in making an extrapolation to the human CNS drug. So the blood-brain barrier has been the major deterrent for the developing drugs. For example, there are some drugs. For example, haloperidol is there. It has a pyridinium ion at the time. It is not toxic to the brain. But if you take the MPTP, the well-known pesticide, it also has a pyridinium ion. But MPTP goes into the brain and gets metabolized by monoamine oxidase to the MPP plus, the highly reactive metabolite, which actually causes the mitochondrial dysfunction in the neuronal cells, which is a major problem in drug development because you don't know which drugs are going to cause toxicities. And in vitro, you can do as much possible, but you need to understand the systematically the drug development in terms of the blood-brain barrier and the toxicities first before you even attempt for the efficacy studies for these molecules, because many drugs are toxic, more toxic in neuronal cells than in the liver cells, because liver cell has a lot of protective antioxidants brain cells don't have. Okay.
I think Dr. Shivan can come in here.
Sorry. Yeah, we could do, but the problem in India is that you don't have the repositories to be able to use this type of cells. So that's one of the major problems in India is that in the US, I can buy these and next day morning, I have any human cell I want on my table. But in India, it's still highly regulated. You can't get the human tissues and cells as yet. We need to open up the market. I mean, any patient died in an accident, any subject who died in an accident on the road, majority of those donate their organs because they can write it on their driver's license that my organs can be donated, and they sign it on the driver's license. They can use that and donate as long as they're free of the HIV, HBV, and other things. If they have those infectious agents, they cannot be used. But otherwise, you can use all those. Many times I got the livers, which were not good for the transplantation. For some reason, they were not. The liver was not able to be transplanted for patients. So they immediately called me. Do you want this liver? They give me for a very nominal price, and I used to take it, make my hepatocytes, and do my drug metabolism studies and all that. That kind of a luxury is not there as yet in India. We need to open up for that kind of research. There are a couple of people are trying to do it, but you're not allowed to take it out of the hospital.
Dr. Shiban, you want to come in?
Yeah. Thank you, Dr. Mangala. It was very informative, very deep knowledge in the subject. I have only one question because most of the physicians in the audience are from background Ayurveda. What's the minimum bar you will set for deciding the toxicity of Ayurvedic drugs in general? The FDA bar is very high. In India, what is the minimum bar you will set?
FDA is working for the patients. It's not working for drug companies. So whatever I say that this is safe, that is not safe, FDA is not going to listen to me. FDA is there to protect the patient. So bar, I mean, I always, because I've always had to excel to reach the bar of the FDA's bar. And that's the way it is. I mean, we don't want to lower that bar. And in fact, I was saying that in 1996, there were 53 drugs approved. By 2000, there were 10 drugs were withdrawn because of the several patients started dying. Then immediately FDA went back to be conservative. That's why you see the valley of death. Because FDA, if they loosen up their regulation and say that okay, we don't have many drugs, let's release more Ayurvedic drugs. But tomorrow they have to answer the public for approving those drugs. So FDA bar is going to be high. It will be always be high. I don't want to be political here, but maybe there are some deregulations may be happening very soon. But it's not good for people to have those deregulations. Okay, that's my answer. But I mean, toxicity, you always have to look at the pharmacology. And as I said, without pharmacokinetic studies, you can never go and estimate your toxicities.
Thank you, Dr. Mangala.
Sure.
Yeah, sir. Shall I take another question from audience?
Yeah.
Yeah, I have a question from Radhika. The question is, what is the criteria for the determination of drug dose to test their toxicity in animal models?
No, there's no criteria here. What we do is we go as high as possible. I mean, I've dosed animals up to 2000 milligram per kilogram. As long as it is absorbed, dose linear, there's no criteria for it. The only thing is that there's always dose limiting toxicity. For example, dose limiting toxicity means it's not the LD50 anymore that people in the back in 1980s, 1970s, people used to consider the LD50 as the benchmark for the dosing. Now, we don't consider that LD50 is not used as totally used. So we actually look at the concentration of the drug in the blood plasma, and that is the deterrent, and making sure that what are the human pathology. For example, if I'm doing animal studies, I have a luxury to be able to take out all the tissues and look at their histopathology and correlate that histopathology with the concentrations that I get from pharmacokinetic data. And that's when I say that this is the concentration at which this on multiple dosing is actually causing some toxicity by the drug. So this is how it's done. There is no hello.
Yes, sir.
Yeah,
Please continue.
we are listening.
Yes, sir.
Okay, so this is how it is done. But the thing is, it's pharmacokinetic based, drug metabolism based, and we have to understand as much as possible from the animal models to. If we are looking at the human safety here, we're not looking for the animal safety here. Animals are only a model to make sure that your drug goes into the humans is safe. Okay.
Okay.
Of course, you have to comply with the ethical concerns of the all the animal against animal research. You know, like PETA, HSI, Society. They have a lot of regulations on how to dose the animals, how to handle the animals, and we have to comply with all those regulations. And that is the way it goes. That's it. I mean, those regulations are binding, and anybody who violates those regulations can be put in jail by the. So you have to follow these animal ethics regulations.
Yes, sir. I have a question like your discussion on drug metabolism was really interesting. Could you please explain a bit more about the pro-drug and its metabolism?
Yeah, you know terfenadine, right? Terfenadine, a drug called it used to be called Seldane. It was a wonderful antihistamine, if I remember correctly. It was antihistamine, I think. So what happened was, at that time, a lot of people were also taking antifungal drugs. Antifungal drugs are very, very potent inhibitors. Ketoconazole is one of the antifungal drugs. It inhibits the CYP3A4 even at less than 1 micromolar concentration. 0.1 micromolar concentration, it can inhibit 100% of the enzyme activity. So people who are taking terfenadine, which is metabolized by the CYP3A4 to the active metabolites. Terfenadine by itself is not active. It's the metabolite, I think it's fexofenadine. I'm trying to jog my memory here. Fexofenadine, I think. And the ketoconazole stops the uh, metabolism to fexofenadine. Now the terfenadine doesn't work, number one. The concentrations of terfenadine go way high up, and terfenadine actually causes cardiac arrhythmias, and many people died because of the terfenadine. Uh, then FDA comes and says that we cannot market this drug anymore because there are too many drugs that interact with this terfenadine. And then they went back and looked at this was, I think, Schering Plough drug at that time. Now Schering Plough is part of Merck. And then when they looked at the metabolite, they thought, okay, metabolite is the active one. Why don't we market the metabolite? This metabolite was already patented by another biotech company. So you also have to look at the patent issues with these metabolites. So if your metabolite is going to be active, if you don't have a patent on it, somebody else has a patent on it, you're screwed. You can't go far anywhere. So metabolite also has to be patented. And this metabolite, for finding what they did was they bought that company or they bought the patent or something. Now fexofenadine is on the market, which doesn't have the same side effects. It is not metabolized by cytochrome P450, and it doesn't interact with the ketoconazole. It's one of the wonderful. I think it's cetirizine right now. So you have to understand this metabolism also very well. And as I said, there are some metabolites that are produced in animals because of animal metabolism is different, and the humans don't produce those metabolites. Yeah, those are animal specific metabolites. But problem comes is when human produces a different metabolite than the animals produce, then you cannot do it. FDA will say that there are some leeway that say that, okay, if the metabolite is less than 10% of the parent exposure in the plasma concentrations, then you may be able to justify as long as the metabolite is not an active compound. Okay, sometimes if you take opioids, right, both the parent drug as well as the metabolites, all of them are active. In such case, I don't know if it is the parent causing the efficacy or the metabolite causing the efficacy. So you need to understand the metabolic pathways very early on. Typically, what we do is we do the in vitro metabolism first using human liver microsomes and compare with the species comparison with the rat liver microsomes, mouse liver microsomes, dog liver microsomes, and see the metabolite profiles are the similar or not. If they are not similar, we're going to kill the project right away because humans are going to be, we will have more, what do you call, FDA is going to stop us from doing clinical trials if there's unusual metabolites or more metabolites in human compared to the animals. We don't want to get into the string of doing toxicology studies with the metabolites. Metabolites may be safe, but FDA is going to ask you to do all the toxicology studies that I described for the regulatory development have to be done with the metabolites as well. Now, some people like in Novartis, what they did actually physiologically based pharmacokinetic model. There's something called SimSip, one of the software tool. They use that and estimated what could be the liver concentrations of the, not the blood concentration. They just estimated what could be the human liver concentrations because they also had rat liver, dog liver, monkey liver data, concentrations data, extrapolated it, estimated it, and showed to the FDA that this is not going to be toxic in liver. And then FDA approved it. They said, okay, you don't have to do the toxicology studies with the metabolite. You can go ahead. So you need to look at how else can you justify to the FDA in such cases.
Thank you, sir. The explanation regarding ProDrug was really in detail. And with your permission, I would like to take a next question. How many drugs which are in the market today since few decades cannot pass FDA imposed safety studies today?
Many of them. Paracetamol will not pass through today with FDA regulations. And it's it's an over-the-counter drug. You can go and buy Dolo. You can go the Crocin and everything. Today, if you have to go to the FDA with as a new drug for paracetamol, it will not be approved by the FDA. Period. So there are so many other drugs like that. FDA will not approve anything before 1980s. That is why people say that it was approved in 1950s. It's been there. There are 5 million prescriptions for this drug. And then I say that the drug development today is totally different from drug development before 1980s. So you have to comply with today's technology. So HCQ is not being taken for the COVID by the FDA. You can say that it was approved in 1950s. It's been there. It's working very well. And but the people on the FDA is saying that it's got too much liabilities, and we don't want it on the market. Period. So that's one of the examples. HCQ is on the market, right? But for another indication, for malaria, lupus, and rheumatoid arthritis, but they don't want to approve the same dosing regimen for the COVID-19. Okay.
Thank you, sir. Thank you very
I
much
can
for
give
your
you a
answer.
lot of list. There are several lists of the compounds like that I can give you.
Okay. Yeah. So thank you for your answer, sir. But as you have said, paracetamol is not eligible to be approved by FDA. And then what exactly is stopping paracetamol to be withdrawn?
Well, see, paracetamol causes toxicity in high doses. It's a predictable toxicity. Predictable means you have to have at least a 3 grams dose minimum per day to be causing liver toxicities and fatal deaths. And nobody takes, and for the pain, so normal pain like headaches and everything, you don't take 3 grams every day. Unless when people commit suicides with it. But that is a different case. But paracetamol is a dose mechanism is very well known, and FDA can control it very well. But whereas if you take the troglodyazone, some other drugs like carbamazepine, it's not dose dependent, right? Many of these drugs that are being withdrawn or also being looked at the FDA to be withdrawn or those drugs, they have a dose-dependent toxicity in the humans. We call them idiosyncratic drug toxins. Idiosyncrasies, we don't know the mechanism,
Okay.
right? So that's why paracetamol can survive now, because we know the mechanism of toxicities, and it is not low dose toxicity, it's a very high dose toxicity. Okay?
Yeah. Sir, I have a next question like, what is your experience with phytomolecules and FDA?
I don't have with FDA any experience on the phytomolecules. Uh, I mean, I have mixed feelings about it. The one problem I have is that the clinical trials so far done for the phytomolecules are not sufficient to be able to make a decision whether they are efficacious or not. I think we need more data, in my opinion, and we need the double-blinded, randomized clinical trials for most of them, for FDA to buy into them. Of course, I mean, many of them you can say these are natural molecules. I can still get them approved by over the counter, but you still don't have the data. You're going with a gut conviction, saying that gut feelings, saying that yeah, I believe that this will work. Uh, so I mean, you need more data. That's my opinion.
Okay, sir. Thank you. Thank you for opinion on phytomolecules. And sir, could you please explain the importance of SIP enzymes and their physiology?
Which one?
SIP enzymes in physiological conditions. Importance of SIP enzymes in physiological conditions.
SIP enzymes are for their primary function are for detoxification of the external drugs or environmental pollutants. The evolution has devised a mechanism to throw away anything in the foreign molecule. Even the drug transporters that are in the gut, intestinal lumen, are to prevent the drugs to get into the body and cause toxicities. And this is why the evolution has actually made the cytochromes P450s. What cytochrome P50 does is it puts a hydroxyl ion. I mean, it also catalyzes many other reactions, but the hydroxylated metabolite is more water soluble than the lipid soluble drug or environmental pollutants. Then water soluble metabolite gets conjugated with glucuronides or sulfates, etc., and gets more water soluble and gets excreted through the kidneys very fast. And this is a mechanism that nature has divided to get rid of the food. But the problem is that nature does make errors, and this is one of the errors. Sometimes cytochrome P50 makes uh, carcinogens and all the other toxic metabolites. Unfortunately, nature is not perfect either.
Yeah.
Right?
Uh, Thank you, sir. And sir, could you please explain the mechanism of action or toxicity associated with amoxicillin plus clavulanic acid, which is known as augmentin today?
Right.
Could you please, yeah.
With the mechanism, as they say that they still don't know what exactly. The speculation is
Mhm.
that it could be forming reactive metabolites. I worked a lot on the reactive metabolites in my PhD thesis and my postdoctoral career, looking at the cancer potential, because there are two things that can happen for reactive metabolites. For environment, most of them environmental pollutants, a reactive electrophilic metabolite that is produced that can covalently damage the proteins on DNA. When it damages the covalently to the DNA, it forms a DNA adduct. Some of these DNA adducts may not be repaired, and that can fix a mutation in it. If that mutation is fixed, that slowly goes into the cancer model, depending on the type of the mutation. Not all mutations will lead to the cancers anyway. And in fact, in most environmental pollutants, we know that they cause cancer uh, because of they form DNA reactive electrophiles. Whereas on the other side, drugs produce soft electrophiles. They're not hard electrophiles. Soft electrophiles can be detoxified very easily. You have glutathione, endogenous antioxidant. You have ascorbic acid, endogenous antioxidant. Vitamin E, endogenous antioxidant. So these antioxidants, if you have in high levels, they can protect you to some extent. But if your metabolism overwhelmed producing these reactive metabolites to a level that these antioxidants no more can be protective, then these reactive metabolites cause toxicity. So there is one publication by Dr. Jack Utrecht from University of Toronto. In fact, I was at University of Toronto. I was actually part of the PhD I did, and I used to interact with him a lot. And he had one theory for the idiosyncratic drug toxicities. What it is that all compounds that produce reactive metabolites may not be toxic. And he said there's a danger hypothesis, and in which he said, troglodyazole, I already mentioned that it is actually within a year, 2,000 patients died. Troglodyazole dose was 600 milligrams at the time for human. And then the rosiglitazone came on board. Rosiglitazone was 25 milligram dose. Whereas the third one, the another sister of that is the pioglitazone came in, Actos they used to call it. Pioglitazone dose was about 7.5 milligrams. Rosiglitazone and troglodyazole survived for too long compared to troglodyazole. And his hypothesis is that drugs doses should not be more than 25 milligrams. So if they produce the reactive metabolites, that overwhelming reactive metabolites that are produced at the high doses can actually cause idiosyncratic immune toxicities. If you look at the amoxicillin clavulanate, what are the doses there? I think 400, 500, something like that, if I remember correctly, right? These are
250.
250, right?
Yeah, clavulanate is 175.
So they are a lot more than 25 milligram dose. So if you're taking daily basis for at least a week, they keep producing the reactive metabolites. They can potentially cause the damage to your proteins in the cells and maybe potentially attack the immune system. Okay, so the dose is very essential. I think it's very hard to generate all drugs at 25 milligram doses. It's important. That's why I say even phytochemicals, you should look for the low doses, not the high doses. Yeah, they may be natural compounds. I said glucose is toxic too. It's a natural compound. Cholesterol is a natural compound. It's also toxic. So natural doesn't mean it's not going to be toxic. It's a dose, but not just the dose. It's a concentration that flips to the toxic level from the pharmacological level. You need to understand what is the differentiating factor between the pharmacology dose and the toxicology dose. If you understand that better, then you can avoid the toxicities that we can see. So the dose plus the concentration, you need to understand better for even for phytochemicals. And for amoxicillin, I would say that the doses are very high, and FDA is now seeing many cases of this cholestasis, which is like a jaundice. My suspicion is that amoxicillin and the clavulanate, they are actually stopping the bile acid secretion from hepatocytes. But bile transporters are there, right, in the liver. These bile transporters can be inhibited by the amoxicillin or other tetracyclines. We know that tetracyclines do inhibit it. I don't know if penicillin types of compounds do that, but tetracycline compounds do inhibit the bile transporters, and that can cause the cholestasis, kind of a jaundice, and can sometimes be fatal. And this jaundice can spill into the hepatocellular necrosis if you continue to have jaundice and you can continue to take this drug, and goes into the liver total failure. So it's called hepatitis. So the cholestasis has got to be controlled very early on, and if there's any symptom that the cholestasis is occurring, then the drug has to be discontinued by the doctor.
Yeah, but amoxicillin clavulanate is a great story about the absorption and how.
What is it? Say it again.
amoxicillin clavinate is a great story about a synergistic activity. But
Yeah,
I think Dr. Shivan can come in here about what you talked about.
yeah. So, okay, I'll tell you one thing. Not all patients react. Okay, any idiosyncratic toxicity happens in only 3 to 5% of the patients. Not all patients react. So, how do you identify those 3 to 5% patients?
Yeah,
If you're
I,
not
I
identifying.
understand. I understand. I request Dr. Ganju to come in here.
Sure.
Dr. Damleji, what do you want me to comment on? The vote of thanks now?
No, what thanks.
This has
Sir,
been too deep and too long. And, just a couple of trivia messages here. Number one, the commonest cause of liver failure among young people in the US is acetaminophen poisoning, which is the active compound paracetamol. The commonest cause of liver failure, the commonest cause of liver getting a liver transplant for young people is paracetamol poisoning. But
You know why? You know why that is?
sorry,
You know why that is? Go back to 1985, look at the FDA.
no, I'm just saying that you're right. There's the commonest cause is there. It's a trivia I'm adding, and that's why it's happened
Sure.
high doses, and therefore it's important to be very careful of the dose also.
Yeah,
sir,
but yeah, let
is
me
it
explain
beyond
it to you
Ray's
why.
syndrome? Ray's syndrome is beyond Ray's
No, no,
syndrome.
not Ray's syndrome. This is just acetaminophen poisoning.
Yeah, I'll explain it to you why. You know what happens? Americans drink heavy, very heavy, and you know the ethanol, alcohol. It is metabolized by cytochrome P52E1 for hydroxyethyl alcohol. Okay, it's also metabolized by the catalase and alcohol dehydrogenase as well. Those forget about catalase and alcohol dehydrogenase. For cytochrome P452E1, alcohol is actually metabolized by it. And actually, alcohol also induces the cytochrome P452E1. Now, people who are taking alcohol every day, the next day, they feel some kind of a hangover, right? Oh, I have a hangover, let me take the paracetamol. You already induced your cytochrome P450U2E1 that metabolizes the paracetamol. Okay, at which produces the toxic quinolamine reactive intermediate. So now alcohol also reduces your antioxidant profiles like glutathione, ascorbic acid, and vitamin E. So there's no protective agent in your liver to protect your. So people who drink excessively, taking paracetamol, are the ones who die of the paracetamol. And this is FDA actually put it onto their warning in 1985, but nobody pays attention to it.
Yes, sir.
Balaji,
Yeah,
questions? Anybody?
Hello,
yeah.
Dr. Madhu.
Hello?
Madhu is here. Madhu is from cortical allergology. You may be having something to comment on this.
Good evening, sir.
Good evening.
Yeah, my question would be about how our research of phyto ingredients in India would be accepted in other countries, especially in America. How is the ground reality happening?
Okay. I mean, I really didn't work much on it. I did publish some of the not really phytochemicals, but I also actually published on ginger, etc., a few papers. But the problem I'm seeing is this: the flavochromarins and other compounds. Is that the problem is that they actually not a single component. There are multiple components. And they all work as a combination very well, not as a single component. That's my bias to it. I mean, I may be wrong. The phytochemicals. The problem is that when you go with a combination, as a drug and you say this is a drug, and FDA is not going to approve unknown moieties, you need to have an active ingredient defined for FDA, without which FDA is not going to accept your phytochemical mixture. Of course, you can say that this people are taking it as a diet or nutrition or something, so it's safe. So I'll put it as an OTC and market it. People do it, but the problem is you're not doing enough clinical trials to be able to really give us not as an allopathic medicine to the patients. I mean, it can be an Ayurvedic medicine without evidence based, but it cannot be an allopathic medicine because you don't have enough evidence to be able to justify it to the FDA. So unless we put more money into it, India is of course strong on the Ayurveda and etc. But we need more money to put it and do more and more clinical trials to show the efficacies that we believe in, that they actually work. And if we could do that, that would be great to do. I mean, that's something we should all do it. I mean, I think I would support it. I would also go to the regulatory agencies if they want to support. I mean, I don't know about DCGI positions here in India, but FDA positions have always been that we don't have an active ingredient here. You need to identify the active ingredient and other things. So we can work with a mixture of chemicals. And that is the problem. Unless you identify all the mixture of chemicals, you can never do it. So we are at a crossroads here. How do we take this forward to convince the regulatory agencies these are better than allopathic medicines? Okay.
Thank you, sir. Long way ahead, it seems.
Yeah.
Dr. Vishnu?
Sir, thank you for your informative talk. Sir, I would like to ask one question. Do we need to study drug interaction for classical Ayurvedic preparations? Like we used to give many classical Ayurvedic drug interaction study.
Absolutely.
Like used to give...
No, this is one of the things I already said, that grapefruit juice killed the people. Right. So the problem is herbal doesn't mean that they're not chemicals. They're also chemicals. The entire body, human body, is a chemical interactions, biochemical interactions. Whether you put as a chemical as a diet or a chemical as a drug, they're chemicals. Both of them can interact with the structure-activity relationships. Cytochrome P53A has some structure, and it attracts some functional groups. And if your herbal compound has some functional groups, it's going to interact with the cytochrome P53 enzyme. So it doesn't mean that herbal doesn't mean it's not going to interact. It will interact, and it has been shown that herbal agents do interact with the allopathic medicines, and there were some serious drug herbal interactions as well. And FDA actually want to regulate that as well, but they still don't know how to regulate it as yet, because these are all on the over-the-counter medicines. And the problem is, I do remember my grandmother one time told me that if you eat egg and you don't eat the snake gourd at the same time, I didn't understand at the time. It just perfectly explains to me that only 3 to 5% people die with snake gourd and egg combination. That's purely due to the chemical interaction of some chemical in the snake gourd with some chemical in the egg is interacting with each other and killing the person. So these are something that its body is a chemicals, and you're putting any chemical into the body, you need to understand the biochemical interactions. That's how I believe. It's biochemistry, chemistry. That's it.
Thank you, sir.
Yeah, thank you, sir. And I have another question. Would it be possible for you to elaborate a bit on your human on chip model?
That's a long way to go, but I'm very over ambitious. Of course, some people say that oh God, you're too ambitious to create this kind of model. But that is where I want to be right now. The human on a chip model, the advantages of that is that they are human tissues, organs, uh, and there's a flow like a blood flow in them through the microfluidic channels and everything. Their advantages is that each animal, I mean, if I'm doing a toxicology study, I need 200 animals for 15 days. I do multiple doses, and I need a kilogram drug. If I have to do a rat and dog toxicology studies, 1 month toxicology studies, I need 2 kilograms of drug. Somebody has to manufacture it, right? So here in microfluidic channels, I don't even need 1 milligram drug. They are human tissues. They are combined. I mean, I'm connecting these tissues in a way that they interact in the human in vivo physiological systems. And I also flow the blood like the blood flows in the human blood vessels with the same blood flow rate and volume, etc. If I can create that kind of an artificial system in this, you can come very close. I mean, I'm not going to say that it looks like a human, but you can create very close to the human system than an animal system that has a different biochemistry. Animals have a different biochemistry. Uh, if you take cytochrome 53, mouse only 70% homology to the human cytochrome 53. So if I put the testosterone in rats CYP3A4, CYP3A enzyme, I have a dozen metabolites from testosterone. Whereas if I put the CYP3A4 in the human and put testosterone, I have only 2 metabolites. Only 6 beta hydroxy metabolite is the major metabolite. So there's a lot of things that are different. Rifampin induces a cytochrome P453 in humans, but does not induce the PXR equivalent of the rat. So rifampin is only a human-specific inducer, not a rat-specific inducer. So you need to understand this biochemistry very well and to understand why animals are not poor. So that's why we want to do more and more human tissues, human models than using the rat dog models. They're good empirical models to be able to get into the safe human dose into the humans. But do they really explain the pharmacology and toxicology very well? That happens in the humans? No, they don't.
Yeah, thank you, sir. And I have one more question. Will it be possible to extrapolate the drug dosage from animal experimentation on the grounds of LD50?
No, LD50 is a concept that is not used in pharmaceutical industry anymore. Because LD50 you're determining by dose. As I already pointed out, it's not the dose, it is the concentration that causes the toxicities in the body.
Okay.
Okay?
Yeah.
So
Yeah.
LD50 is useless project, and FDA will not allow you to go to the LD50 approach. They want your toxicokinetic data extrapolated to the humans from different animals.
Yeah. Thank you, sir. And could you please explain the Happerton hypothesis of immunotoxicity?
Yeah, that's you're talking about drug-induced immunotoxicity, right?
Yeah.
Okay. This is $1 million question. There are lots of hypotheses. Uh, Here's my pet one. My pet hypothesis is this. I've already described you the genetic polymorphisms of cytochrome P52D6. Right? Now, if you look at the immune system, in either you're talking about the adaptive immune system or innate immune system, especially the cytokine exaggeration, take the IL-6, IL-17, TNF alpha, IL beta. For example, IL-6, right? IL-6 gene actually has a mutation in the gene promoter region, not in the actual coding region, that actually inactivates the gene IL-6 is not produced. So like that, there are also a lot of pro-inflammatory and anti-inflammatory cytokines have a lot of gene polymorphisms. Not only, there's also toll receptors, human leukocyte antigen receptors. Majority of them have gene polymorphisms. And if we sequence all of us in this gathering right now, we will find lots of polymorphisms, and we'll all be susceptible for idiosyncratic toxicity for not the same drug, maybe different drugs, depending on how we dispose the drug. So my feeling is that this immunotoxicity is coming from the genetic polymorphisms. But the final thing is that you also have antibody, what do you call these? Antigen, antibody, antibodies against the antibodies, right? So what you have is autoimmunity questions. So IL-17 beta, there's actually in rheumatoid arthritis, they've already described that there is IL-17 beta, which is actually the therapeutic agent, that the antibodies are produced against IL-17 beta. The IL-17 beta doesn't function. The people get into the idiosyncratic immune toxicities. So there's antibodies against these natural products are some of the reasons why you have immunotoxicities as well. So these are the two hypotheses that I have, that you have genetic polymorphisms on one end, and also the natural molecule. For example, I said reactive metabolites are forming in hapten, right, with some critical protein. Maybe it's an IL-17 it is binding to, right? The IL-17 peptide, it probably has some sulfhydryl groups, the covalently attached to it. So that activates immune response by itself, because that's also damaged. So there will be antibodies against that IL-17. So what you're creating is a antibodies against your natural products. For example, anti-nuclear antibodies are the major reason for the fatal lupus erythematosus. So anti-nuclear antibodies, not all lupus patients have them. Some people have them, and they have to be extremely careful because they have antibodies against their own DNA. So these mechanisms are still not understood very well, but there are lots of them are at hypothesis level still. Okay, I'll leave it at that.
Thank you, sir. Thank you very much. May I now request Dr. Shiban, sir, to render the vote of thanks?
Dr. Wangla and all the participants, thank you for coming. Dr. Wangla, you're awesome. You're so detailed and thorough. It was a pleasure listening to you. I like to listen to you many more times.
Thank you. Thank you very much.
And then all the participants, thank you for coming, and thank you for staying there for this long. It's been the longest session today, almost 2 hours, and it just shows your interest and patience. And Dr. Damraji, thank you. Dr. Lata, thank you. And Dr. Bharat, thank you for moderating every time. You're just awesome with the background arrangement.
Yeah.
And we'll be having more of these sessions in future. Please join us and please invite your friends so we can get bigger classrooms than what we have today. Thank you very much.
Thank you all the panel members. I'm really happy with all the very provoking questions, very interesting questions. And Rishi, you haven't asked any question, but I really thank you from bottom of my heart for giving me this opportunity. And hopefully we'll interact more and more together. Okay. Hello.
If there's nothing else, can we close the session now? If there's nothing else.
Yeah.
Yes. All right.
Yeah.
We see you all next time.
Thank you.
And we'll see you all next time. Wait for the next announcement. Thank you very much and good night.
Good night.
Thank you. Thank you very much. Good night, all.
Good night.
Thanks, everyone. Good night.
Good night.